Okay, I think we'll get started. It's my remarkable honor and pleasure to welcome back Mark Hlatky. Mark is actually a remarkable individual. He did his training at Penn. He did his residency at University of Arizona, then UCSF, and then to Duke, and went on faculty at Duke, and then now has stayed on after leaving successful starting of the data bank efforts, then went off to Stanford and has remained there ever since. He leads up his own group there and currently heads also an AHA Outcomes Training Center grant, which is the thing that brought him out here today. He gave a great talk last night where he discussed the relative values of angioplasty versus bypass surgery and survived a mixed audience, and now is taking on the world of biomarkers. I think by his description, this is an interactive talk, which I warned him maybe isn't the right answer with this crowd, but we'll definitely interact with you some on challenging evaluation of biomarkers.
Thanks. Do I need to do something, or is this on? It's on? Okay. Yeah. Well, it is great to be here and look out and see a lot of familiar faces in the audience, and it's nice and glad to see everybody. And I hope I can give you some interesting insights into this whole problem of evaluating novel risk markers. I got to get used to where I can see here and see the slides, and you see me at the same time. So. This is going to be oriented more towards risk markers as risk markers for developing disease, but I just want to point out that the same framework applies to markers in people who have established disease, and we're trying to figure out whether they're helpful in managing people who have existing disease, either coronary disease or heart failure or whatever. But the framework is related to prevention right now. So with that caveat, I'll just go through this. And some of this is a very mixed audience, and there's a lot of people for whom a lot of this stuff is going to be old hat, and there's a lot of people for whom it's totally new. And I think it actually works a little better if it's, you have a question, stick your hand up, and we'll try and address it earlier rather than later. So the main thing, we have this whole we have this whole concept of risk factors for developing coronary disease, classic Framingham study characteristics. They established this whole idea of risk factors, and you're all aware of the classic markers, age and sex, cholesterol levels, whether you smoke or not, blood pressure, diabetes, are all well-established things for developing something called the Framingham risk score and estimating what the chances is somebody will develop heart disease. And there are other markers, too, that don't necessarily make it into this score but are well known, like family history, genetics, physical activity levels, obesity, and BMI measures, and so on. The big bottom line, though, is a lot of the risk for coronary disease is not well understood, and we're not doing a great job at predicting who's going to develop disease, even with all of these risk markers. And so there's a great interest in doing a better job of improving risk markers, which is why we're trying to motivate this. So what use is it to identify something as a risk factor or a risk marker? I think there's two things. You can use this information to help develop population-level interventions to say, well, we as a country need to do something about obesity. We as a country ought to put less salt in the diet, things like that, less fast food, whatever. And we can for that we don't need to measure things in individuals. We just need the insight from the biomarker or the risk marker studies to recommend those kinds of things. But we also need to make treatment decisions for individuals to help them decide what to do. And our preventive measures can be behavioral change, which, as we know, is the hard thing, or the easy thing, which is to recommend drug treatments for these various things for like blood pressure or diabetes, et cetera. So there's a lot of interest in doing a better job at improving on Framingham risk markers, the Framingham score or other existing risk markers. And we're trying to detect the disease process or subclinical disease. And there's a lot of new technology around this that is developing these new risk markers from advanced, hit the wrong button here, imaging, We now have coronary calcium scores, carotid intimal medial thickness scores. We can do this. They send a truck around that does these mobile screenings, and we can all have this stuff done. There's new biomarkers. A new one is coming out every week. We have, CRP levels, LP, LA2, et cetera. Lots of things that are in the pipeline. And one of the reasons I got interested in this is because of genetics. And, we heard a lot of hype about how genetics is going to help us. We're just going to need to measure some new markers. And so the question is, how do we evaluate these markers? How do we keep score in terms of figuring out which ones are useful or not? And so this is a paper that we published from the American Heart Association Scientific Statement, which I was the chair of this committee. And the focus of this was basically to say we had a committee on novel risk markers, and one of the biggest questions was, well, how do we evaluate and how do we keep score? And we tried to make this as independent of any specific marker as possible, so it was a general framework. And the idea was that we wanted to have this be a little like the consort statement for trials or other things as a reporting standard. So I see Penny, I told Penny, we want the American Heart Journal and other journals like Circulation and so on to adopt this and apply it whenever you get a new paper to come in. Because we all see, one of my chief complaints is you see this paper and it's like, God almighty, they didn't do any of the stuff that we recommend. How are we still publishing these things? So maybe I need to work with the editors group to see about, getting these kinds of things more adopted by the editors and saying, you need to use this checklist. But ultimately, of course, the idea is if you change the reporting, you're trying to change and improve the design of the studies that you're doing. So I hope in the end, we'll do better studies of these risk markers. So one of the things to say is that I've used this as a risk marker talk, but it's important to say that every test that we might come up with could be used for different purposes. We can use this to make a diagnosis. We can use it to assess risk or prognosis, and we can use it to select treatments for patients. And, exactly the standards we apply might be different depending on the use that we're going to apply it to. And this actually comes up in a big way because a lot of these markers might be thought of as diagnostic tests, or they might be thought of as prognostic tests. And I would say that the biggest argument in the committee, or one of the discussions we had, is this issue because a lot of the way that people think about these markers are related to diagnosis. And I would argue that we actually need them for prognosis and risk, and we need a different intellectual framework. And the biggest thing is that, in a diagnostic test, we can detect right now, today, whether the person has disease or not by some gold standard test. Therefore, we can use cross-sectional designs, and the right ways to think about it are sensitivity or specificity of the test and predictive value. And a lot of the stuff on risk markers is couched this way. But in fact, we really need to be doing prognostic studies because outcomes then occurring over the future. So we need cohort designs, and the right ways to quantify it are maybe more hazard ratios and relative risks or odds ratios or things like that, rather than sensitivity and specificity. So I'm going to go through these things, but this is, if you take home nothing else from this talk, this is probably the main thing, is to say that we have put forward kind of successive layers of evaluation, of stages of evaluation of a novel risk marker, and I'll go through these individually. And actually, I'll say that what I'm going to do in the talk, and I encourage questions, is go through these, and then at the end, and if there's enough time, I'll apply them to the case of CRP testing. So the main thing is to say we first start with statistical criteria. So we look at those and we apply those, univariate association, multivariate modeling. I see a lot of our statistical colleagues are here, and I'm glad for that, and I hope you'll ask questions. The next part, though, is what I would say I would put them as decisional criteria, which goes with risk reclassification measures and assessments of how we change treatment as a result of these things. The next level beyond decisional criteria are outcome criteria. As a result of doing this, using this test, measuring this risk marker, do we do better management? And as a result of the better management, do we have better clinical outcomes? And finally, because this is what I do, I'm interested in the cost effectiveness. Okay, well, maybe the outcomes are better, but is it at an affordable price? Is it good value for the money that we're spending? So each one of these are successive criteria that you would apply in evaluating a risk marker. So let's start at the beginning. Statistical recommendations, and this is really the easiest in some sense to say, we really need to have some ways of looking at this in a statistical sense. Whenever you report a study, tell us about the basic study and where the patients came from, what their outcomes were. Tell us about easy things first. Tell us about their standard levels of risk markers. And for the new marker, tell us something about relative risk, the p-value, and confidence limits. We ought to be knowing what the p-value is for stepwise addition of this novel marker to a model that includes the existing things. And I can't tell you how often we see this where people just put in the new marker and say it's highly statistically significant, and they don't consider simple things like age and sex. And it seems kind of silly to do that. We also want to have reporting here of the model, the discrimination, like the C-index, measures of discrimination, and measures of accuracy of the risk marker and the risk predictions. And I feel at this stage of the talk I'm totally bringing coals to Newcastle, but I'll go on and tell you, because this is a lot of stuff I learned when I was here. Our key points of concern, though, were that whatever was done should be transparently reported. And I got back to that thing that saying that we wanted to affect journal editors. So Dan, in the back, you can look up and say that you're supposed to use these criteria in the American Heart Journal. So we want to apply these kind of things. We want to have full and transparent reporting of risk markers. I'm glad you're here, Dan. Thanks for coming. How much the new marker adds to the simple ones, and one of the basic, the incremental part, oops, I keep hitting the wrong button when I mean to hit the laser pointer. How much is the incremental value of these things is critical to this. And the other thing to say is we felt that there was no single statistical measure that captured everything we were interested in. And the idea is if no single measure is good enough for everything, we ought to be reporting multiple domains of this about any one of these measures, so not relying on just a single one. And so how good is the risk score? We know that there are these two concepts, and I learned this from Kerry and Frank back when I was a fellow here. That we need to think about separately the idea of accuracy, saying that if we have a 10% risk, predicted risk, we want the risk to be 10% in the real population, because if it's too high or too low, we'll make the wrong decisions, especially about interventions to prevent things. And the second thing is that, if we predict 10% risk for everybody, we'd be highly accurate but not very helpful, because we haven't made any discrimination between the people who develop events and people who don't. So we want to test a score or a measure that is highly discriminating and also accurate. And, this is the concept of this, and I must say I'm generally PowerPoint challenged, so this was like a huge success for me to be able to put together something like this. But anyway, we have here, you want to see this in a risk marker paper, and we often don't, where we just actually plot how good they are and predicted and observed over some range. Here it was deciles. But the other thing we want to know is how discriminating it is, and the C-index is very popular, and Duke had a lot to do with popularizing this. We wrote a paper a long time ago in JAMA that said that this was a really good measurement for diagnostic tests, and you probably all can explain this better than I can, but I'll explain it anyway. That the idea behind this is if you pick two random people out of the population and one of them had an event and one of them didn't, that the person who had the event has a higher score, risk score, than the person who didn't. And so if you were just randomly picking people, in these pairs, you'd be 50%. So 0.5 is really bad. And if you were 100%, you'd get 1, so that's perfect. So this is the C-index, the area under the ROC curve is another name for the same thing. And, here it is graphically, again, with my highly sophisticated drawing, showing you that, here, if you randomly picked this guy and this guy would be higher, but if you pick this guy and this guy, they would not. Whereas in this thing here, the C-index is very good because no matter which one of these guys you pick, they had a higher score than all of these guys. So that's a very good discrimination, whereas this one is okay but not great. And actually, for those of you who look at C-indexes, 0.84 is actually pretty good. And you can see that overlap is actually a fair bit. So, they're even with a relatively high C-index, we still can get things that are pretty much overlapped. So, to move from the first part from these statistical criteria to the second one criteria are kind of decisional criteria. How do we use the information? And this is the border zone between those two things, because there's new measures of trying to say, well, we need to try and get at how much things are changing as a result of using a new risk marker. And so the idea is that if you have a risk marker and you have a predicted risk in a guy who ultimately develops an event or disease, and it moved that individual's estimated risk up, that's good. And if it moved the estimated risk down, that's bad. Very simple-minded. And the reverse idea is on the other side. If somebody didn't develop things, you want that risk marker to move them down and not up. And so there are new measures that try and see whether people have been moved in the right direction or the wrong direction. And we try and do this in a statistical way, and there's a description of this in Statistics in Medicine. Mike Pensina and others have developed two ways of looking at this. Which is to see whether you've moved in the right direction by any amount. So people just move; it's just qualitative. They go up or down the right way. And the second one is, well, maybe you moved everybody by up by 1/1000th of a unit of probability. You'd get a really good thing, but it may be useless because you didn't move them very far. Did you move them far enough that you would change their estimated risk in a category that's meaningful? So that's the Net Reclassification Improvement Index. They applied this in an interesting paper that said, well, let's evaluate this brand new marker, HDL, using these criteria. Because we all are saying, well, HDL is a pretty good marker. We really do measure it. We believe it's helpful. And they looked at the Framingham Offspring data for 10 years of follow-up. And, of course, HDL has a hazard ratio that is protective. Good cholesterol, very significant. But the C-index in this model, actually, if you add HDL to a model that doesn't have HDL and you add HDL to it, the C-index improves by only a smidgen, and it isn't statistically significant. And so a lot of people complain that the C-index, but the problem is that there's kind of a ceiling effect. We can only get so good at predicting things like coronary disease events, which maybe have some inherent unpredictability about them. So maybe we're not capturing the full thing. But if you apply these new metrics to it, this integrated discrimination index, this number doesn't look very big, but it's quite significant. So it moving up a fair number of people, and this reclassification index is actually quite significant. And here's the idea and the fundamental data that shows this. Let's see if I can walk you through this. This laser pointer doesn't show very well here. Can anybody see this when I'm in the back? You can? Okay. So this is the percentage of people who are in a risk category, and these are risk categories that are generally accepted, ATP3 levels, to say under 6%, 6 to 20 is intermediate, and over 20%. So here's the people who were in that category before using an HDL, and here's the category that they were in after they had the HDL test. And so people who are on the diagonal stayed in the same category. So there was no movement. And the ones we're interested in are those people who are in the off diagonals. So this top row here is the number of people who experienced a CHD event. So these are the people you want to move up, right? So moving them up is the right direction. And so these highlighted things, yellow, are the right direction. You can see there's 15 people who moved from low risk to intermediate risk, and 14 people who moved from intermediate risk to high risk. But counterbalancing that, there are some people who moved in the wrong direction. So 4 people moved down from intermediate to low who had an event, and 3 moved down from high to intermediate. But if you compare the two, there's 29 here who moved up in the right way and 7 who moved down in the wrong way. Well, that's good. Okay. And here down on the people who didn't develop an event, you can see that they're reversed, right? The ones that moved down are the right direction, and the ones that moved up are the wrong direction. So these are yellow here. And the number of people who move down and the number of people who move up, if you add them up, 173 people were reclassified up, 174 down. Really, it didn't change anything very much. So by this metric, this is why it does well in this net reclassification index, because there are more people who moved up than down in the case group. That makes sense? I mean, this is kind of, many of you may know this already, but maybe not. But, you know, it's intuitively relatively simple to see, especially, and one of the things we say is people ought to publish data like this using the new risk markers to say we need a table like this or a plot that helps us understand what's going on in this way. When you add a novel risk marker, you know, maybe we all believe HDL is really good, but what about, you know, some fancy new marker that Kristen Newby is putting together? I think I saw her here. So, you know, I mean, how are we going to believe whether, you know, we trust her, but we're going to cut the cards? We're going to say what is it that makes that a really good marker or not? So the idea is that if we're evaluating new risk markers, we ought to move to this decisional level of criteria to say, how are we using this information in practice? Any of these statistical measures don't necessarily measure action criteria. We don't necessarily know how the information is used. But if we do these reclassification measures at generally accepted action thresholds, say, okay, you know, by the way, everybody agrees that if you're more than 10% risk, you ought to get started on this drug. So we use 10%. We don't use 15%. We use 10, which is something that will be tied to decisional criteria. And in prevention, in cardiovascular disease at least, you know, there are a number of reasonably well-accepted criteria. So this works pretty well in the prevention framework to use these reclassification measures if you tie it to accepted thresholds. If they're tied to arbitrary thresholds, it's maybe less helpful, you know, if you just made them up, especially if they were data-derived, because you picked the threshold that makes your marker looks good. So, you know, it's better if they're designed beforehand. So let me get into this idea again, but just by doing this from a decision theory framework, which is to say if you didn't have a test available, you would have two choices. You would decide either to treat somebody or not to treat them. And there's some, if risk is going up as you move across this thing from left to right, there's some threshold, some number above which you would treat people. That's your treatment threshold. But, you know, if you had a new test available, you could change that. You know, you could fiddle with this. Maybe you could also add a test and then decide who to treat. So the concept is that there's a treatment if you have a testing threshold as well, and that, you know, we ought to set things up this way that there is a zone in which we should test people. And exactly where you draw those lines depend on how good your treatment is and how risky and costly it is and how informative your test is. But the concept here is that really super-duper low-risk people, you still wouldn't do the risk marker because chances were they'd never move above a risk threshold. If you're super high risk, you would still treat them even without doing it because you're worried about a false negative or moving them too much in the wrong direction. And in the middle here, you'd test people. So a lot of recommendations are you test intermediate risk people because those are the people you're most concerned about. Okay, so we're moving through the framework for evaluating novel risk markers. There was purely statistical criteria first, decisional criteria second. The third thing is that, well, okay, we want outcome criteria, because maybe what we're doing is we're just shuffling the deck. We just change who we treat, but nobody's any better off for doing it, except the people who made the test. So, you know, we need to decide whether, in fact, all of this testing is actually leading to better outcomes, which would include things like mortality and morbidity or quality of life. But the thing to say is that most of these tests that we're considering have very few direct effects on these outcomes. They may, in fact, you know, maybe you'll feel bad if you learn you have a bad gene, and maybe that's something we need to consider. But for the most part, you don't have a general thing about this, or maybe you will change your behavior because you heard you have higher risk. But given how terrible we are at changing behavior, I'm not sure that this will work this way. Most of the effects of testing are from treatment changes. So did we change treatment at all? So the decisional criteria were, did we change what we do? But the second one is, okay, you changed what you did, but are people better? Maybe you made stupid decisions. Like you get a coronary calcium scan and you end up doing cabbage and everything. Sorry, Bob. But, maybe that's a bad idea. So, you changed what you did, but for the worse. So you need to know what the outcomes are and whether they're improved or not. And that's actually a big issue and controversy, I think, in the field, because a lot of people who are doing diagnostic tests like imaging are saying, well, what is this? I mean, you're raising the bar for us. I mean, we shouldn't have to prove that our outcomes are better. I just read in the paper yesterday a whole big thing about CT colonography, whether we're doing, it's a good idea. And all the manufacturers, the FDA is saying, well, we've always approved tests just based on diagnostic criteria. The fact that maybe we're hurting people by doing these tests, we can't take that into consideration. But it's raising the bar. I would argue that they need to raise the bar. And, we need to figure out better ways to evaluate these things. And the final thing is to say, okay, well, after the dust settles, is this strategy of testing a good use of money? Because some of these tests cost a lot of money and some of them cost a little money. And we can apply cost-effectiveness analysis to a testing strategy as well as a treatment. So in the usual cost-effectiveness way, we compare the test strategy with the next best strategy, and we look at how much costs is added and what the added effects are. But the crucial insight here and importance is that the cost of the test is an intention-to-test type strategy. So if I order a CRP and then I start somebody on a whole lifetime of medications, the cost of that testing strategy is not the cost of the CRP test, but the cost of CRP plus all those drugs I ordered or all those further tests I did. So that we have to account for all of those additional costs, all the downstream costs. And the effectiveness, again, is based on clinical outcomes. And this is done very, very, very infrequently for tests. We don't do much cost-effectiveness analysis of tests. So those are the different ways that we go and look and apply these different criteria. I thought it was interesting, and we've put in the paper just this idea of how this happens chronologically. Here we are in the DCRI. You guys are thinking up all kinds of great thoughts about what would be a new risk marker. You first are going to go through and develop this risk marker and follow a number of stages. You're going to do some studies that are proof of concept to show, well, hey, it actually is higher than people who have heart attacks than people who don't. Then you're going to do prospective validation studies to prove that this marker really does work and assess the incremental value of that marker compared with standard established markers. So that's very, very important. And then we're going to have to look at these stages here, clinical utility of those, how much we've changed therapy, and how much outcomes are changed. And we can even consider doing randomized trials of marker-driven strategies. And, there is some precedent for doing that, like we have had randomized trials of abdominal aortic aneurysm screening and so on. And I know people are trying to organize a trial of CT, coronary calcium scanning, and, other kinds of risk marker evaluation. So trials are a good idea. But, most of the time we're stuck here in these first early few stages, and only now are a few markers getting to this intermediate stage where we're actually looking at incremental value. Most of the time, all the stuff we read about is up there at the first or second stage. So before I go on, I want to apply all this stuff to CRP. So I'll walk you through the CRP. Before I get there, does anybody have a question or a quibble or something? Dan.
Value, which is of critical importance in this. Sometimes I see a lot of papers where people who are trying to do this rigorously will test a new marker against the absolute best model that exists so far. Mm-hmm. But that test is a different evaluation than the question of what does this marker do in the clinic when you've got a clinician with a brain that is processing information completely differently, deciding what the next test is to use. Just like your decision-making thing concept before, should we be, it's interesting to know that if you throw every possible variable that you have into the model and test the incremental value of some new thing on top of all that stuff, that the answer is X, but should there also be some test that says if the average clinician practicing with the average amount of information that they're able to process, which is a couple of items of risk data, uses this information, are they going to make a better decision with it, regardless of what they could do if they were using mathematical formulas and precise predictions, which they're not likely to do right now?
Well, you know, it's an important and interesting observation and question. You know, what you're really saying is that we often don't have good decision support systems for people to get them the information they need, and, you know, adding two numbers together is too complicated, and actually doing the Framingham risk thing is laborious, and maybe we need a way to make that information readily available to people, and that just doing that would improve decisions immensely, maybe more than all of these other risk markers. I mean, you're saying we're not even getting the standard stuff into the clinic, let alone the fancy stuff. You know, I would argue that, you know, you probably ought to fully get the other stuff out to where people are using it in a better way first. But, you know, that's a good question. So you're saying, you know, there's no added value, but, you know, if it encapsulates all the value that's there in a single number, why not let people use that number? You know, I think that's that. I mean, so it's not incremental to a good model, but it's incremental to a lousy model, which is the clinician's brain, right? Is that what you're saying?
Precisely.
Okay.
But
Well,
it also improve care.
Yeah.
Yeah, I mean, Dan, to put some numbers on that, it's estimated that the Framingham risk score mark is calculated 6% of the time
Mm-hmm.
of primary care practice in adults in whom that might apply.
6% of the time.
6% of the time.
So
with something that's, you know, a great score is not being used because it's laborious to do.
Right. Well, I mean, I would say the big opportunity, I know I hear a lot of hype about electronic medical records, but this is sort of low-hanging fruit of saying, you know, if your computer screen can give you this information right away when somebody walks in, you know, that would be a really good use of electronic systems and better decision support to give us what we need at the time of the point of care.
So a great test isn't being used because we are too dumb to do it or because we don't get paid to do it. If we could do an imaging study, we do it pretty consistently without a problem there. We could do 1.2 echoes per patient per admission to do, in part because we get paid a fair amount of money to get that done. So should we either A, reimburse differently, or B, otherwise reward the need to calculate these risk scores?
Well, I mean, I think it's both. I think that we're getting paid to do things, and so we do the things we get paid for. Cardiology in particular, we like to look at pictures. So we order pictures. We're less interested in, you know, adding stuff up and beyond sort of three-vessel disease. But it's an interesting, you know, it's an interesting concept. If you take, for example, the TIMI risk score, which we all love to bash, but one of the, you know, its C-index for non-fatal infarction is what, like 0.64 or something. It's not very good, but it's easy to do. I mean, you basically have to be able to count to 7 to create it, which is about the range of what a cardiologist can do. So, you know, there is a trade-off of simplicity versus precision.
Well, the other thing that was commented on is, you know, we're not paid to think, we're paid to do things. And, you know, I would argue that's another thing we need to fix.
So if we got paid $10 to calculate the Framingham risk score, Eric, you think
You'd
that
see
would...
it.
Charge for it, yeah,
Absolutely.
a CPT code or something. There you go. You'd probably see all kinds of use for it.
So another question, Mark.
Make the fellas.
These Framingham risk score is a prime example of this. It looks at sort of 10-year risk, which in part is majorly determined by just how old you are. The idea that if you're treating in a primary care clinic 30-year-olds, nothing affects their 10-year risk, which is kind of good. You can have a lot of risk factors, but it has almost no impact on your risk profile for the next 10 years because you're young. You're not going to die of cardiac disease no matter no matter what. The degree to which we need to change these scores to something like lifetime risk that are out there also, as an idea of seeing if you continue to smoke or have high cholesterol, its impact isn't going to be seen in the next decade if you're young. It's going to be seen over that lifetime.
Well, you're absolutely right about that, and I think that one of the things that this points out, though, is if we're going to evaluate these things relative to outcomes and treatments, it depends on how good our treatments are and how risky they are or not. Your threshold for recommending that somebody stop smoking is essentially zero, because there's all the good evidence is that it would be good. Whereas, saying, by the way, you should lose weight and exercise more, that's probably okay. But, to start a drug that maybe you're going to take for 50 years, that's, there's a different question about that. And, you might say, well, golly, I don't know if I want to do that. My concern is that today we spend far too much time. It's a lot easier to write a prescription than it is to get somebody to change their behavior. So, what happens is we get a bunch of sedentary, overweight 30-year-olds who smoke, and we say, here, take Crestor. So did I just go over the CME thing? I mentioned it. So anyway, that's the problem. Carrie.
Yeah, I would just emphasize, Mark, as you went through some of the statistical steps there, the univariate assessment of the relationship, and what does this add to other factors that we already know about. I think all too often people just throw information about a new risk factor or a biomarker into a model without the kind of careful assessment as to the nature of the relationship with the outcome and the shape of that relationship and so on, because that can really, as we all know, affect one's interpretation as to how much it might increase the C-index or improve the accuracy. So it's embedded in those simple steps in your slide are some key steps that a good statistician would always take in terms of assessing the nature and the shape of those relationships in order to model it correctly.
I agree with you, and in fact, when I think back again to some of the target audience for this stuff is reviewers and journal editors for papers of touting new markers, that we want to make sure that they pay attention to that step and that they really are using the information. The caveat of Dan's comment, well taken, that clinicians are not necessarily using that, but I think if the claim is you're doing better than you could with other information, you need to use the information in the best way. But the Framingham thing is a fairly standard score, so there is a way to do that. Although that is fairly non-controversial, where you're not building a model on your own. So, all right, let me take this specifically to CRP for a minute, because I think actually the whole CRP thing kind of was one of the things that led to this committee, I think, being formed, because there was a lot of controversy about it. A lot of people who like CRP were saying, wow, the measures that you guys are using are terrible because CRP looks bad. And other people were saying, well, we shouldn't use those measures because it makes CRP look good or whatever. Our stance was I didn't care where it fell out. I mean, what we wanted was things that would apply to any new marker and not just to CRP or coronary calcium, whatever your favorite marker is. Or a new biomarker signature or whatever. So just applying these criteria in a successive fashion, where does CRP stack up on this? And as far as the statistical criteria, I think I'm ready to give it to them, to CRP, and say, by most of these standard statistical criteria that I laid out, there's a lot of evidence that says CRP is pretty good. Here's a meta-analysis that was published and showing from pooled data the effect of looking at different quartiles of CRP versus relative risk of developing coronary disease, and a number of studies have done that shown that it is incremental to Framingham risk score in a statistical way. So that's been pretty well proven. What about reclassification? This is something that came out last year in CV outcomes, where people applied CRP levels to a Framingham, I think it's the Framingham population. I didn't put that on the slide. And what you can see, actually, interestingly enough, is a lot of people stayed right here on the diagonal in terms of their risk assessment. But, some people were moved up and some people were moved down. This is actually not presented in the kind of way that I would like, where you break people out as to whether they developed an event or not. So you can assess these new accuracy scores. But there is some reclassification that goes on, and you can see that most of the reclassification is in the people who were intermediate to start with, which says that there's some targeting that maybe would be valuable in terms of using the CRP. What about outcome criteria? Are people better off for using this? And here, I think, is where we're running off the road a little bit myself, because CRP is an inflammatory marker, but we're not rationally treating people with anti-inflammatory drugs. We're mostly dealing with them with other kinds of interventions. And, we don't need a CRP level to tell people to lose weight and stop smoking, nor is it really going to change treatment of diabetes and so on. So it probably isn't going to change that. The main place it's used right now in terms of the focus has all been on whether to initiate statins or not. And, my own view about this is that statins have been amply demonstrated as effective in a lot of trials in a lot of different patient populations. And if you look at all the data from all the studies, it pretty much is, in relative risk terms, reduces risk across the board in a proportionate kind of way. And so the expectation is, no matter how you pick the people, it probably is going to reduce their risk a little bit by maybe 20%. The Jupiter trial got a lot of press. They selected patients, and this is a very large study, where people were selected in part because they had a high CRP and an LDL that was below conventional criteria for starting statins. And they did show here a highly statistically significant risk reduction. So that, the way I look at this, is more evidence for statins in lower risk people. What it was not, though, is a trial of whether CRP was useful in deciding who to treat, because everybody who got into the study had a high CRP by design. If you really wanted to figure out how good CRP was, you would randomize the people with low CRP as well and see whether it's a predictor or not. But that was not what was done. So, this is preaching to the choir again here. I think everybody in this room knows about this whole idea of proportionate and relative risk actions for therapies. And, for the same relative risk reduction, you might get more bang for the buck in certain categories in high-risk patients than in low-risk patients. You reduce your risk by 0.2, you go from 20% to 16%, or 5% to 4%. It's pretty much the same. The key thing to look for, though, is whenever a marker is an effect modifier. In other words, it is identifying people who respond to treatment versus those who don't, like a relative risk of 0.6 in the overall 0.8 in the overall population might come because half the people respond really well and the other half don't respond at all. So if you have a marker that tells you which half is going to respond, that's a very important thing to know. Or perhaps it's not so black and white, but there is a little bit. Respond in people who are positive, the relative risk reduction is 0.7 versus 0.9. And this would be treated as tested as a treatment interaction with this covariate. This is extremely important for patient selection and whether this is cost effective. And if you think in another field, we look at cancer therapies, and there's a lot of things that say, well, here's a molecular marker for breast cancer. And if you have this marker, you respond to this treatment, and if you don't have the marker, you don't. So, hey, this is going to determine which treatment you get. Do we have things like that in cardiology? I mean, maybe there is a little bit. People who are troponin positive seem to get most of the benefit in acute coronary syndromes from a lot of interventions compared to people who are troponin negative. So they are really critical to find out whether is there evidence that CRP is such a marker? Now, if you listen to Paul Ritker and read the papers, you'll get the answer that the AFCAPS, TEXCAPS showed that this was true. Actually, if you look at the data, this is a slide you won't find from the New England Journal because you have to calculate this yourself. So I put this up just to show it, which is in that paper, they looked at people who had high or low. This is a large study of people who had statin treatment to prevent heart disease, the average CRP was about 1.6 and the LDL was pretty high. And they had stored samples. So they fished these out of the freezer and then they measured CRPs and correlated that with the response, which is a nice design for a biomarker study. So it's a very good way to do this, at least if your biomarker is stable when you freeze it. And the thing that they reported and focused on was in people who had low LDL cholesterol and a high CRP, they seemed to get a really good risk reduction, whereas people with a low CRP and low LDL did not. And this got a lot of press, and they make a big deal out of this, and this is the whole rationale for Jupiter. But actually, if you back calculate the rest of the table, what you find is that the group that did absolutely best was the group with low CRP and high LDL if you treated them with statins. There's sort of when I look at this, I say this doesn't make a whole lot of sense for saying this is a treatment a response indicator. And in fact, they report in this paper that the interaction test for CRP with LDL treatment is not conventionally significant. So even in this kind of discovery sample, it wasn't a significant result. So I'm still waiting to see the data that says that CRP is in fact an indicator of treatment response to statins. And my prior on this is very low. I don't think it is because if you look at things like obesity and diabetes, statin treatment reduces risk proportionately in all of those groups, and they're associated with higher CRP levels. So my guess is that CRP is just a garden variety risk marker. It doesn't predict who really responds to treatment in a big way. So how does this play into the CRP cost effectiveness? One of the things that's important to look at is that, a lot of, actually I should need to update this slide. 3 recent cost effectiveness analyses say that if you take lower risk patients and treat them with statins, that it's pretty cost effective if the statin is cheap and it's really quite safe. So if you buy that it's cheap and safe, you probably ought to broaden the treatment criteria. And if you accept this, is it better, what's the right strategy? To go back to my slide that says, there's a window there for which you would test people before deciding what to treat them. And if you actually do, you can test this, you can say, well, the strategy is to test with CRP and treat people, or just treat them. And it turns out if you do the analysis, it's really very narrow window for CRP testing. We've written a paper on this and it's under review but not yet published. So there's actually not a lot of reason, in my view, to do CRP testing in a lot of people. It's only really when you need to make a decision. But I do think that if it turns out that it is a risk marker in the way that was described and it predicts who responds better, if it really does work that way, then it will turn out that window is much wider. But there's really no data right now to say that that's the case. So if I were to look at CRP in the framework that we had, I'd say it meets the statistical criteria. It probably meets decisional criteria because we apply these over risk thresholds, and it does reclassify people. But there's not a lot of evidence yet that using it is going to improve outcomes or that it's a cost-effective thing to do. So I wouldn't recommend doing it widely, but I do think it can be very helpful when we're on a close call with whether to treat somebody or not. And I think one of the interesting things that I would test is whether it might have an effect in patient adherence to treatment, because one of the other things that is that people who really should take statins don't. And if a risk marker were to motivate people to change their behavior and adhere more, then it might be valuable too. But we don't have any evidence yet for that a lot of these risk markers affect what people do in terms of their behavior. Novel risk markers, I think we need more of them. We need to really do improve prediction. A lot of this is just driven by new technology development. But I really think we do need to do this critical assessment, and maybe I need to work with Dan and some of the other editors to make sure that we apply these kind of criteria that were developed for new papers that come out. So we're doing this in a better way. We really do need to assess incremental information and look at what we're doing about decisions. But in the end, the main thing is to make sure that by using these tests, we're actually improving outcomes and not just kind of shuffling things around. So I'll stop here and answer any additional questions. Dan.
Actually, I sent you a recent paper on biomarkers that you declined to review. So how am I supposed to improve if you won't even help me out? I was
So bad he didn't even want to review.
actually, he had reviewed it once. It was just a re-review to see if he liked the improvement. But
It's probably just in a cranky mood. I'm sorry.
you were traveling and you weren't going to be able to do it in a timely fashion. How's that?
Okay.
So I was thinking about our president's recent physical examination results, which were widely disseminated, and the response of some of the cardiology community that was quoted. And I remember, I can't remember who this was. It was definitely not somebody from Duke, but I think it was somebody from New York who responded that Obama's LDL would have definitely made him start statin therapy, except for the fact that his high sensitivity CRP was low, so he felt that he didn't need to do that, which struck me as embodying two interesting, potentially wrong pieces of thinking about novel risk markers. One is he was looking at the LDL only, and then two is he was allowing the high sensitivity CRP to dominate the LDL information. And I'm just curious as to whether or not, since these are like expert people who are being asked by the media to give opinions on this, whether we're really in a position to actually make intelligent decisions about patients using novel risk markers.
Well, I think we have a way to go. Actually, the first thing I'd say is that he has to stop sneaking out back for a smoke first. That's probably the thing that I want him to do first. But yeah, I think it's a way to go. And one of the things about it is that there is a bit of confusion, and I didn't really mention this, but there's a huge commercial interest. A lot of these things are being pushed by some company or another that either direct to patients or pushing this in some other way. And so these financial interests tend to also push us to want to do these things, and they get promulgated more than would be reasonable or rational. But I think you're right that people are not necessarily thinking about this in a kind of coherent, reasonable way. Yeah.
One of your early slides, you mentioned that you got interested in this because of genetics.
Yeah.
How do you start thinking about the complexity of the so-called genomic signatures as opposed to a single risk marker? Are we to think of the criteria you laid out as applying to the genomic signature?
I didn't put that in there, but we did have a section in the paper to deal with the signature issue because, I've been involved with some people who do this, and one of the things that I found is that they're forever tweaking the signature. So, you get the signature, and it works real well. You find the optimal signature in this one patient group, and then you do it in another one, and you say, well, it didn't come out so good. But, if I adjust this and adjust that, it looks great. So, this is still a great signature. And saying that kind of a thing is not a good idea. And one of the issues for a lot of these signatures is you can't find out exactly what went into them. Like I'm on a panel where we were looking at breast cancer genetic markers, and the algorithms are proprietary. You cannot actually find out what they're doing with the marker set. It's a total black box. So one of the things that we said was that we should be more transparent about what's going on, and the models need to be stable, for us to evaluate this. These kind of multi-marker things, there's overfitting is a huge problem, and we need to validate using the initial way it was done, and not, allow people to continually fiddle around with it to change how it's going to come out.
That's an interesting comment locally because Duke is spinning out companies based on the ability to come up with proprietary algorithms and establish signatures, and now we're beginning to advertise for patients to come to Duke and get your genomic signature figured out, and we'll treat you on the basis of that. So there are some real issues that you're bringing up.
Well, I went in on a project where, I was convinced by all the genetic things that, gosh, this is going to be great. And I went in and we did a whole study on this, and I must say I was disappointed that, we came up with so little in terms of predictors for this. So, I think it's a lot tougher problem than people are making out. And absolutely, it's more than one gene marker is going to be it, but we need to have ways of looking at it that are reasonable. So, this kind of need to be proprietary on the one hand is in conflict with the idea of being transparent about it. Well, thank you.
Okay. Thank you, Mark.